So thank you so much for the, oh yeah, continue. The recording started. Okay, let's just start. Yeah, Tayana just introduced me very well. So as she said, I've been working in developing methods to bridge the mechanistic toxicology with ecological effects. And this motivation has started more over than 10 years ago. And I focus on this, and I think this is important because we need to improve the tools that prevent irreparable damage in the marine ecosystem. So I just want to start high pointing what an ecotoxicology is, or what we aim to do. So we study the harmful effects of chemical, biological, and physical agents on living organisms. And our objective is to improve environmental protection. But basically, we want to answer two impossible questions, which are, which of the many thousands of chemicals poses the greatest risk to the environment, and which of the millions of species in the environment are at higher risk? And to do so, this being two main approaches. The first approach focuses on using bioassays of cellular or molecular markers, and promises an upscaling to higher biological levels. And this can be possible for the implementation of the adverse outcome pathways, but it's hard to extrapolate to different species, because we know that there are mechanisms that are species specific at the molecular and cellular level. The other approach is chemical stress ecology, and treats pollution as another of the many factors or variables that has an effect on the species and the populations. On one hand, this approach aims to provide ecological realism to the environmental risk assessment. And on the other hand, it fails to discriminate if the effects that we are observing are due to one single contaminant or to the mixture of them. A way to extrapolate to different species with the AOP method is implementing depth models. So here I'm just giving you some concepts and theories, and I just want to introduce depth theory. It stands for dynamic energy budget theory, and it was developed by Bas Kochman in the late 70s. And it's a theory that describes how available energy fuels physiological processes. What is interesting about it is that it defines how these budgets can vary over the life cycle of an individual, and how they are affected dynamically by the temperature and the food, which are the main forcing variables of the model. This theory is applicable to all organisms, and basically the species will differ on the parameter values. I will come back later to my presentation to depth, and I will give you more details about it. Right now, I just want to describe the pathway that I follow to realize that this is a very good tool for extrapolation in ecotoxicology. So my interest in ecotoxicology started during my bachelor in Venezuela back in 2006, where I studied effects of contaminated sediments on enzymes on a bivalve. And by then it was clear for me that studying a single species will not be enough to protect the marine environment. Then I have the opportunity to join the Census of Marine Life initiative. The project was basically marine ecology. And this was great for me because I saw from the ecotoxicological perspective that we usually focus on single species or that are called the sentinel species. And this there's a blurry line between it's not a blurry line, actually. I think it's a very steep separation between the eco and then the toxicology. So we used to forget the eco part. And during my time at the LEA lab and this project, I have the opportunity to reinforce my ecological theory and thinking, and biostatistics. And I co-authored these 3 publications. Then it was clear for me that I wanted to pursue an academic path. So I moved to Europe to do a master. It was an European master, and that's why I have been in so many countries, because during my studies, I have to spend 1 semester in each different country. And for my project, I work with mullets, which are fish that we find in the estuaries here in Europe. I don't think you have them in Australia. And these mullets, they start showing a high occurrence of intersets. So my objective and my work aim to standardize the expression of different genes involved in esterogenesis, and to link this expression of these genes to the occurrence of intersex fish. Then I did my PhD in Norway in collaboration with the Center of Marine Studies in Brazil, and I will talk further in the next slide. And right now I'm in France. And I will talk to you about this also. So my PhD aimed to develop biology-based tools for predicting risk and impacts associated with diesel oil spills. I'm showing you here a framework that was proposed by Gotzel, in which he was actually talking about bioindicators, and I used the analogy with biomarkers. And what was funny is that I discovered this framework while I was writing my thesis, so at the end. And I noticed that all the different experiments that we planned and we did in the thesis fitted well in each of the categories. So the objective is actually to go stepwise by establishing first correlation and cause and response to determine which are the markers or the indicators in this case that allow for monitoring studies, that are useful for monitoring studies. So I conducted several experiments, and they're all summarized in 5 peer-reviewed publications. And the first 2, I look for establishing first the correlation and then cause of exposure of diesel, first in different species, 5 different species. So I have a baseline of different species that we found in the area of study and how that changed the biomarker response. I work with antioxidant enzymes, how these antioxidant enzymes responded to different seasons. And then I tuned a bit the design and choose 2 of the previous species and did another experiment in the lab. And then the third step was to check for response. And response means or relates to studying the sensibility due to different doses and frequencies of the stress factor, in our case, spills of diesel. Then I also explore the differences in species sensitivities when exposed to a single PAH. So in this presentation, I will talk to you about these 2 papers, these 2 last papers. So the first story is a monitoring story. So it's the 3rd paper that we did for proving that antioxidant enzymes could be used for monitoring diesel contamination. And what was interesting about this study is that we did it in the field, and we use low concentrations of diesel that we spill directly in the mudflats. And the interest of using low concentrations was to replicate what the species were exposed to in real life, because the area of study is a mudflat that is in a channel that goes towards the harbor in Brazil. It's actually the 2nd biggest harbor of the country, and so there's a lot of traffic of ships of an important size that comes in and out the estuary, and are recurrently spilling low concentrations of diesel, or this was our assumption. So we wanted to check if the antioxidant biomarker activity changed after exposure in the field, and if the biomarkers were capable to show us a short-term response and a long-term response. So for that, we designed an experiment that consisted in 8 quadrants in the field that were exposed and separated for control quadrants for at least 80 meters distance. And so we have 2 blocks to see if there was a spatial variability. And we didn't bring the animals. The animals were already there. So we just checked if there were enough amounts of these Bibles that I didn't have a picture to show you, but it's a Bible. And of course, one of the important assumptions of the experiment is that the Bibles will not move more than the area of the quadrant. So this illustrates a bit what we did. So we went to the field and we placed these aluminum square frames. And on day 0, we spilled on the quadrants that were meant for exposure. And we came back to the field 7 days after and 14 days after. That didn't, and we sample animals. So that's what was to check the response. 48 hours after exposure and 1 week, and then 2 weeks. And after collecting on the 2nd week, we spill again. So there's low concentration, frequent and recurrent exposure was what we were evaluating. At the same time, we have control replicates that didn't have anything that were undisturbed, and we just collect animals in every collecting time. Because there's no time independency on the data, because we collect all the time in the same place, and because our controls are also in the field and potentially subjected to many other all the abiotic and biotic variations that we can find in the field. The way that we analyze the data was adopting the log response radius. So these are calculated for each endpoint at each sampling time. And what we use them is for measuring the effect of the experimental manipulation and assuming that all the variables are the same for both control and exposure quadrants. So the figure, what we have to read is that if the confidence intervals are crossing the line at 0, it means that there are not significant differences of that point at that day regarding their control. So in a nutshell, for GST and digestive glands of the Bible, we saw that they have a response 2 days after each spill, in which the enzyme was you don't say downregulated, it was inhibited. The activity was inhibited. And what was nice to see was that the response was consistent for the 3 different spills. So we did this in digestive glands and gills, and I'm just showing you the digestive gland results. And as I just said, what the highlight of the results is that the enzymes do give us a response 48 hours after the spill, and we can see it in different other markers. And there's usually a recovery to control levels 1 to 2 weeks after exposure. So this is the end of the monitoring story now, but I just want to highlight that this is the last piece of the puzzle. So we saw in the field that the markers were responsive to different frequencies of exposure, and that was really exciting. So our recommendations were to use different specific markers for this individual and measure in different in specific tissues. So moving on to the second paper. Here explore the differences of all these species that are distributed in different regions to a single toxic polyaromatic hydrocarbon, which was the 2-methylnaphthalene. The driving question was to determine if there are regional differences between the species and the exposure. And this is because in risk assessment, what is usually done for regions that don't have enough data, like the subtropical areas or at least in Latin America, is to use the surrogate approach. So we use the metrics obtained from temperate species, and we assume that there's a similar sensitivity to the compound. And want also to check if the SSD approach, so it's the species sensitivity distribution curves, identify these differences. So to do so, we use a toxicokinetic toxicodynamic model, which is the General Unified Threshold Model of Survival. For now on, I will just say GUTS. And this model has, in this simplest version, has only 4 parameters. And for that, we have to get survival data. So what we had to do was to expose 26 different species to our compound, and we quantify survival over time. And then we have a toxicokinetic model that defines the speed of the uptake and elimination of the chemical, and a toxicodynamic model that describes the effect of this chemical inside the animal. So the parameters are the scale damage, that is for the kinetics, and for the dynamics, we have the background hazard, which is a parameter that describes the deaths that occur and are not related to the chemical exposure, and the hazard rate. And then finally, the hazard rate is how fast the animal dies. So how fast is the effect of the compound. And finally, a threshold concentration parameter, which we call the no effect concentration. And this parameter is what we use as a metric for risk assessment. Is similar to an LC50, only that is independent of the time of exposure of the experiment. So LC50s can vary for species, and the same species and the same chemical combination can have different LC50s depending on the length of the exposure of the experiment from which they were derived. And that, in theory at least, doesn't happen to NEC values because these are independent from the time of exposure. So the objective, as I just said, was to get these NEC values and to build the species sensitivity curves from different regions, Arctic, temperate, and subtropical regions, and to see if there were correlations within the parameters that could allow for prediction and hopefully extrapolation to untested species. So these are the results that we got for the SSDs, and dash line are the hazard concentration for 5% of the species. So we saw there was a bit of difference between, if I don't say anything wrong, the subtropics have higher tolerance than Arctic and temperate. But as you can see, the curves are pretty much overlap. And this separation these differences in the HT5s are driven by only one species that is an Arctic species that is very, very sensitive to the exposure. We did this curves for different the characteristics, including the feeding yields. And in all cases, we have curves that were pretty much overlap. So far from our results, we could see, yes, we can use the temperate species as surrogate for subtropicals because there are not differences in the sensitivities. However, being myself, the person that made the experiments and looking at how the rate and the velocity at which the animals were dying, the subtropical animals were dying, I was a bit unhappy with this result because I felt there was some part of the story missing. So I thought at least that the complexity of the species is not well represented by these curves, and decided to look further on my data. And what I looked was for an alternative way to check on sensitivity, and this was given by another parameter of my model, and is a TK parameter So what we checked was the velocity to look at an effect. How fast we can start looking how fast we will reach the next value, and how fast will the species start dying. So this graph shows you the how fast the average. Sorry, yeah. So how this is the average of in time that takes for our species to start dying if we increase a 5% the external concentration regarding their necks. So each species has a neck value, and if we increase the exposure concentration, external concentration of 5% higher to their neck, they will start dying. In this case, the Arctic species start dying 2 days after the exposure, whereas temperate can withstand over 9 days and subtropical over 7 days. With this information, we also decided to do a categorization, what we call the sensitivity categories. So we basically rank or we sort the different species regarding their neck values and the time to die or time to effect values. And we came out with this. So we see that, for instance, mollusks and trophic level 2 species are overall more resistant to methylnaphthalene than the other species. So in summary, TK-TD models are useful, and when we include different parameters or multiple indicators, we get a better story or a better grasp of what is happening. And yes, they have a lot of potential applicabilities, including extrapolating to different species and checking for patterns regarding the chemical nature of the component that they're exposed to. So yeah, now my PhD is over. Now what? And that was like the very, we have all this. We all go through this moment of we don't know what to do. So coming back to my second slide, where I was talking about the bridge, I just revised which was the status of my bridge. So I have so far working at least 3 projects that focus solely in bioassays of cellular or molecular markers, and on other 4 projects that try to combine these markers with chemical stress ecology. But I think I thought that was doing a good job. But the problem that I saw is that the bridge that I was building was too species specific. So I was really orbiting around the species Tivella mactroides, this bivalve from subtropical areas. And this was not the goal. And why I'm so obsessed about this bridge, again, because it's important to link effects from different levels of biological organization to be able to prevent damage at the population and community level. And the question that we have to ask ourselves all the time is, can we really extrapolate? And most of the time we have to say no, because many biotics and abiotic factors do influence the effect of pollutants, and then there is too much uncertainty. And extrapolating basically is not straightforward, because we don't have a theory that allowed us to link the effects of toxicants at molecular cell and cellular levels to changes in life history traits. So this is an example that came in a Roman Square paper, and that actually, I think, changed my life, because illustrates pretty easily the problem that we face in ecotoxicology. So in physical chemistry, we can describe a macroscopic event, which is the perfect gas and the Boyle's law, system, by linking it with what is happening at a molecular scale, and is the kinetic model of gases. There's a theory that allowed us to link these 2 processes, these two observations, these 2 different levels of organization. in ecotoxicology, we are missing that theory. We do have models that explain what is happening at the molecular cellular level. But then when we want to go to higher organization levels, we don't have that theory. Or we are not working enough on that theory, because the theory does exist and it's a depth theory. So I said that I was going to talk to you more about depth theory. And so basically, as I just said, I said at the beginning of my presentation, it explains how energy that full physiological processes is sent to different processes. In that theory, we have four variables, which are reserves, structure, maturity, and reproduction buffer. So when we know this, we know everything we need to know about the individual, but they're very hard to quantify. So we focus more in the processes that feed this variable. So key processes are assimilation, digestion, mobilization, growth and development, and then reproduction. And what is nice is that we can study how this energy is attributed to different processes along the life cycle. So for instance, an embryo that does not feed has reserves that are mobilized for growing and feed somatic maintenance and the structure. So the animal starts growing. Once they start feeding, so we have an extra energy input that has to be assimilated, that goes to reserves, and then it can be mobilized for growth and maintenance. And then there's another part that starts feeding the maturity. Once it reaches puberty and maturity is completely filled, we have an adult individual that starts allocating what was before for maturity, start allocated to a reproduction buffer. So it's like a box that once filled is emptied, and it's emptied in the form of eggs or offspring. In that theory, key concepts are surface and volume ratios, because the energy that is assimilated depends on the surface and volume relationship. And all these processes are summarized in only 14 parameters. There are multiple applications for depth models, and what is nice is that it has been applied already for over 2,800 species, and then most of the cases the model fit well the data. It has real life applications in conservation, aquaculture, general ecology, and ecotoxicology. And it allows for interspecies comparison when we compare the parameter values. There are important implications for management and conservation, among others, and has brought new insights about biodiversity, physiology, and ecology. In ecotoxicology, probably the most interesting aspect of that is that it allows for deciphering the physiological mode of action, which refers to how a chemical stress, single of mixture, affects the energy allocation in an organism, and then consequently the life history traits. And this is a key element for extrapolating from small to larger biological levels. So for instance, if the exposure, usually we can see in juveniles or in embryos, is that after they're exposed to a chemical stress, the energy that is channel for growth has to be a part of that channel for detoxification processes. So exposed animals will grow less than unexposed ones. So these are the 5 different types of physiological mode of actions that can be depicted with depth models. So following my PhD and knowing all this depth story, I decided to write a proposal for funding a project here in the Université de Bordeaux. And my project aims to understand the synergistic impact of climate change and contaminant exposure on the flatfish Solea solea. And the way we will do this is by modeling the energetic cost that this combined exposure has on the fish larvae. So flatfish and the Solea solea are interesting species because the benthonic adult animals that live in the continental shelf, and that after receiving a temperature stimuli, they spawn in, and these eggs hatch and undergo metamorphosis and migration from the continental shelf to, again, nursery areas. And all this process, which is quite complex, is dependent on temperature and hydrodynamic, food availability, and the whole metamorphosis process is quite sensitive, to small changes in what they're used to. Moreover, once they are in the estuaries, they change from pelagic to benthonic lifestyle, and they, depending on the condition of the nursery, which means what are the food conditions and the temperature and then levels of pollution in our case, they undergo recruitment. The salts are an important food eat item in Europe. They have not been able, the aquaculture industry haven't been able to make them grow in aquaculture facilities. And so as they are benthonic and they eat benthic animals, they tend to bioaccumulate contaminants associated with the sediments. So these are all the different characteristics that push us to work with this species. So these are the objectives of my project. Well, first we started by reviewing the literature, getting data on physiology growth, metamorphosis success, and all this data allowed us to calibrate the depth model. And then once the model was calibrated and we can run predictions, we move on the experimental part of the project, which was to produce data for validating the model predictions. So the first part is done, and now, now we are working on the experiment. So the experiment is ongoing right now. And as you just said, the objective is to validate the predictions and also to add what I call here a TD module to depth, and that's called in the literature a depth touch model. And depth touch model will allow us, if we are lucky, to identify the physiological mode of action of the exposure scenario. So I'm being very vague about the contaminant that we use, and that's because we changed it from the beginning. We aimed to work with PBCs, and then we changed to PFOS. And for the experiments, we focus on larvae because it's easier to work with larvae. And the way we are doing it is to place different larvae in wells of a microplate and monitor the development and the metamorphosis success a long time at different concentrations of PFOS. So before running the current experiment, we did a pilot back in 2019. And so these are some pictures to illustrate what we are trying to do. So here you're supposed to see a tiny little larvae. Here there's one, but I'll show you a video in the next slide. And so here is the same individual that grew along our experiment and didn't reach complete metamorphosis by 28 days post-hatching, but was pretty close to it. But what was nice about this pilot experiment is that we quantify what feeding at libitum or to satiation means. And this is an assumption of that model, that the growth that we are observing and is always assumed to be under ab libitum conditions. And what we did was counting the amount of food we gave to each fish and how many of those artemia, artemia is what we fed the fish, so it's a crustacean, how many of those artemias were eaten every day. So here we have on the left a larvae that is on stage 2 of metamorphosis. So it just start to move the left eye, and I'm giving them food. So I'm counting and giving them very carefully each artemia, and you see how they starts eating. And on the right you have it's not the same fish, but it's later on it's a stage 3, almost full metamorphosis larvae. So it's already flat and has already the left eye on the top of the body, but it's not complete. It's not finished the migration. And well, they're both looking happy and eating during the experiment. So the data we got from this experiment was really rare. We didn't have this in the literature. And that allowed me to collaborate with Florence Munier, who was working also in a depth model for this species, but her focus was more on bioaccumulation. So I gave her my data, and we worked together to calibrate the depth model with the data she had from the literature and the data I produced. So on the top of these two figures are the feedings of my data. And what we measure is biometrics, basically, is the dry weight of the larvae along time and the physical length. And so you have the same thing, but for juveniles and adults. And here's annual egg production. So Florence finished the model, and the results were quite satisfying. And she also made these predictions for the larvae stage, where she overlapped my results with predictions of what ab libitum and high food quality means. So it's the gray lines, because obviously in nature they just don't eat artemias, these larvae, they eat morphine. So we are restricting the diet of the individuals to a single species, which is not necessarily what they need. So basically what we saw is that our larvae were well fed. They were growing as expected, or kind of as expected, between what is high food quality and low food quality, but they're not were fed ab libitum, because they were over here. This is predictions. So with this information, we decided we tuned our experiment design, and for the current experiment I'm doing, we have 4 different groups. So an experimental group, which has plates with larvae inside, and that we will not touch. We will just monitor their survival and metamorphosis. And I'll say we will not touch because in my pilot, I monitored the size and the weight of the size, not the weight, the size of the same species along the same individual, sorry, a long time. But that demanded translocated the individual from the plate to the binocular, and that was they were not so happy, but they didn't die about it. But I was worried that that added to the temperature stress and the chemical stress will be too much for my larvae to withstand. So we have a group that is fought for biometric sampling. So we sacrifice the larvae along every week to monitor size and weight. So the results of the specs are will be used for, as I said before, to validate the predictions and to build this TOTS module. And if we are lucky in the long term, we can also do an interspecies comparison with another species that we can find here in France, that is Soleus senegalensis, but its distribution is more towards the south and the Mediterranean. And the hypothesis behind is that the geographical distribution might change in future years because of climate change, because there are more warm climate species. But yeah, this is not for right now. So I'll show you just preliminary results from our experiments. This is survival a long time, and in blue we have the data from optimal temperature and the different treatments. So we have control, and then we had different concentrations of PFAS from low to high. And what you can see is that even in the control and the optimal temperature, we have a lot of mortality. So there's after 7 days, we have more than 50% mortalities in all treatments, including control. So this was not the case in my pilot experiment, which I just have 2 deaths. And so we think there are multiple reasons because of what happened here. One is that the larvae were not in the best condition from the beginning. And then the other thing is that we missed the window for the first feeding of the larvae. And they need to have a specific stimuli of a lot of prey and a specific day. Otherwise, they don't eat. And that's what happened here. They didn't eat. So we repeat the experiment and we make sure to give them a lot of food on the right day, and the survival rates are much better. And the other thing that we did is that we allowed the larvae at 20 degrees to acclimate to the temperature, because in the previous experiment we just put them on 20 degrees right away, and of course that was a thermic shock and they were not happy about it and they died very fast. So this is ongoing, and these are preliminary results. They haven't started metamorphosing yet, or I think my student said to me yesterday that at least at 20 degrees they start looking different. So maybe they are shifting from stage 1 to 2, but the whole metamorphosis can last 6 weeks. So we are in the middle of the experiment, and I hope that we don't have all the larvae dead by then. So to summarize on my postdoc, I started in January 2019. I did a literature revision and calibrated the model using larvae data. And that allowed me to move on the experiment. And with the data of the experiment, I will validate the model, and I will bring or attach a touch module to the depth model. And the next steps that I don't think it will be covered with my postdoc is to couple this model with a bioaccumulation model. So these are the publications that could go out of each of these categories. So to finish, what is the future perspective of my work, and why it's important to study the effects of temperature and contamination in larvae ontogenesis? And is, if we think in different disciplines, fishers in ecology are worried about the future stocks and the geographical distribution of the species, and if it will be changed because of climate change, or if there will be time shift between the spawning and the availability of food for the larvae, and then impact the survival of individuals and the community in the population, of course, and have an effect on the population. In ecotoxicology, there's a good link between the effects of contaminants and molecular and cellular levels, but when we want to go on population levels, we don't have a tool to extrapolate, and it's even worse if we want to go up to the community level. In aquaculture, we're interested in checking how survival and growth can increase, because that will increase profits, and how adapt the methods for rearing the species in the lab, so we get the biggest profit. And environmental chemistry are interested in linking concentrations in the environment with bioaccumulation in the animals, and tuning the methods to I forgot the word, sorry, to quantify at a higher resolution the concentrations in the environment. So depth models can help all of this, or solving these problems or these questions, as they do provide in fisheries and ecology, for instance, they provide insights about the effect of temperature and food levels on growth and reproductive success. And we could also link or couple depth with larval dispersal and hydrodynamic models to make predictions about the time and size of the fish at spawning and the larval distribution and link it to recruitment. And regarding species biogeography, the models coupled with habitat maps could give us a understanding of how species distribution can shift, could or will shift in future climate change scenarios. In aquaculture, we can check if antibiotics, probiotic, and disinfection methods are beneficial for growth, for instance, or survival. And in environmental chemistry, if we couple depth models with bioaccumulation models, we can provide, this will provide means to predict the dilution effect due to growth and estimate also the concentration of chemicals ingested and the effects that that has on growth of the individual. In ecotoxicology, the data that we have from molecular and cellular data and our analysis bioassays is still useful because it allowed us to interpret the physiological mode of action and also to build the AOPs and of course understand the mechanisms of toxicity. But once the interest of depth is that could give us this physiological mode of actions that has a direct link with life cycle traits. And these measures at the individual level are easier to extrapolate because there's the theory that allows for linking individual to population and community. It already exists, it's ecological theory. And moreover, the outputs of depth can be inputs for population models. So that's what makes it very interesting. And finally, if we want to check on different species or extrapolate between different species, we can compare the parameter values and check for patterns between across species. Actually, similar species have similar parameter values. So sometimes if you don't have the parameters values for your study species, you can use a phylogenetically similar species and work with that. So wrapping up, we have these two impossible questions and these two approaches. And I think that actually the bridge between these two approaches is, as I said in my second slide, using AOPs and depth. And yeah, impossible is nothing. So I hope I give you a grasp of what I've been trying to do in these past few years, and that you find it interesting. And I just want to thank you for your attention and for inviting me to this seminar series. It was very nice to wake up violently to science on a Wednesday morning. Thank you.
Well done, Adri. That was really good. Thank you again for such a great presentation. That was, wow, yeah. You went all the way from antioxidant biomarkers to a theoretical model to trying to finally understand everything. Before we finish, do we have questions for Dr. Sardi? You can also type in if you're not comfortable saying it.
I have here the relevant links from that I cited in my presentation, and also the references if you want to look at it afterwards.
Oh, great. Yeah. I tag you into the divulgation of the seminar as well. I should have asked you before if you could, because in this way, if someone has a question, they can actually just send it to you. Do you have a question about the depth theory? Do you know if those depth models they're being already used for coastal management? And if not, do you see like good potential for it, or we are too far away from it yet?
Coastal management in which sense?
For the pollution and how that would affect the ecosystem, or even like provision of functioning.
Yeah, so I know in soil ecotoxicology they are being used aggressively in the last years. So basically in the private sector, so they do these GOTS and depth models to determine the thresholds of toxicity that are allowed. And that's more in the sense of replacing already existing metrics, LC50 metrics and stuff like that, for more robust metrics for risk assessment. Here in Europe, the is being accepted by the European Commission to accepted and slash is a way to invite researchers to work further on that. The models that have been, let's see, stamped as good to work with are the survival model, so the GOTS model, and DEB is still not completely accepted, but it's because it's complex, because of its complexity. So what they argue is that if they want to produce a good data set of effects of contaminants in different species, you have to undergo a lot of preparation and studying to actually be able to work with DEP. Whereas with GOTS is a model that has 4 parameters versus a model of 14 parameters. There are already tools in the internet in which you just have to put your data and the modeling is done automatically and gives you the parameters and everything. So in that sense, yeah, there are studies. They're not, I mean, it's increasing in quantity, I'll say, the research on this.
There is hope.
Yes, there's hope. But you know, like it's an interdisciplinary work, because you will need a depth modeler to calibrate the model for the study species. Then you will need an ecotoxicologist, and if you're lucky, your depth modeler is already an ecotoxicologist, to give some insight and understanding of why you want to study this or that contaminant in that situation, in that species, and to interpret the results of the physiological mode of action and couple it with the mechanistic toxicology information. And then if you want to go on actually in the coastal area, you need another person, probably ecologists that do population modeling, that use the data that you generate from DEP to put it inside the population model, and to say, yeah, actually, if this species is exposed to this concentration and this temperature and food scenarios, we have this physiological mode of action that impacts the population at this, and this level.
Yeah, for sure.
So it's a teamwork, and it's basically my interest in doing these seminars. Of course, there's always interest in talking about your research, but it's also to spread the gospel, let's say it like that, because this theory has been out there for a while now, but there are not so many people that know about it. And I think their applications are really, really, a lot of applications are interested for many different disciplines. So that's why I try to illustrate in my last 2 slides, not only in my small niche of soil larvae, there are many other applications that are not related with ecotoxicology. And then doing the effort to work with DEP will help different disciplines Well, that's my opinion.
Thank you. Well, thank you again for your presentation and for waking up so early to share your knowledge with us. And thank you everyone for joining our seminar. We should have another one in 2 weeks. See ya.
Bye. Thank you again.